Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Using Real-World Data in Single-Arm Clinical Trials When It's Done, Why It's Done, and How It's Done. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Sineos Health, who developed the content for this presentation. Sineos Health is the only fully integrated biopharmaceutical solutions organization. The company, including a contract research organization and contract commercial organization, is purpose-built to accelerate customer performance to address modern market realities. They bring together approximately 24,000 clinical and commercial minds with the ability to support customers in more than 110 countries. Together, they share insights, use the latest technologies, and apply advanced business practices to speed their customers' delivery of important therapies to patients. Now it's my pleasure to introduce you to your speakers for today's event. And first, I'd like to introduce you to David Thompson, Senior Vice President, Real World Research with Sineos Health. David is a health economist who has been working in the field of real-world research since 1988. Among various current external affiliations, Dave is a steering committee member for the Clinical Trials Transformation Initiative, CTTI, a multi-stakeholder initiative of Duke University and the U.S. Food and Drug Administration, a team leader on the CTTI initiatives on real-world evidence and pragmatic clinical trials, and an advisory committee member for the Duke Margolis RWE Collaborative. And next, I'd like to introduce you to Dr. Craig White, Senior Vice President and General Manager of Outcome Science with Concert AI. Craig is a health policy researcher with approximately 20 years' experience in healthcare data and analytics. Craig's research is broad and has covered value-based pharmaceutical pricing methodologies, innovative uses of RWD in clinical development and traditional health economic models, and cost-effectiveness analyses. Dr. White's research has included evaluation of thyroid and prostate cancer treatment during his fellowship at Massachusetts General Hospital in the Program for Cancer Outcomes Research and methodological work as a part of the NSF NIH-funded Bioinformatics Research Training Program at Harvard Medical School. Dr. White received his PhD in health policy from Harvard University and BE in electrical and electronic engineering from the University of Auckland. Now, before I pass over the controls to our first speaker, I'm going to launch our first poll question, and you should see it appear in front of you right now, and there it is. I'll give you about 45 seconds to complete this poll question, and the question is, coming into this webinar, how much knowledge of using real-world data for creation of external control arms do you have? So please select one of the below. And your options are none, I'm starting at square one, a little, but I have yet to dig in seriously, some, but I have not yet had an opportunity to apply it, or a lot, I have been directly involved in execution of it. So please go ahead and cast your votes, then I will share the results with you, and then we'll start our presentation with David. Okay, so thank you everyone for that. It looks like we have the majority of our audience members have cast their votes. I'll leave another 10 seconds for the remainder to go ahead and do so now. Okay, thank you everyone for doing that. Now I'm going to close the polls and share the results. Okay, so here are the results of our first poll question. And the question was, coming into this webinar, how much knowledge of using real-world data for creation of external control arms do you have? And we have 26% that said none. I'm starting at square one. We have 32% at a little, but I have yet to dig in seriously. We have 32% again, some, but I have not yet had an opportunity to apply it. And 9% at a lot. I have been directly involved in the execution of it. So thank you everyone for participating in that. And now we're going to pass over the controls to our first speaker, who is David. So David, when you are ready, you may begin.
Okay, thanks very much, Sonia, and these are really interesting results. And thanks everyone for attending. Thanks everyone for contributing. Right off the bat, we're collecting data here on your experience, and it's good. We've got kind of across the board results. Those who are already experts, there are fewer in numbers, which is good, but we'll expect some advanced questions from that 9% of you who responded a lot. The good news for those of you who are coming into this with a clean slate, the 26% of you who responded none, we have some very fundamental materials here that will help you understand the topic and become a good foundation to build on. And for those of you who are in the middle, a lot of material as well directed towards you. So thank you again for joining us, and I'm going to get right into it. There are three themes that I want to cover before passing the ball over to Craig. The first is the recognition that the critical role that regulatory is playing in all of this, that the FDA and other regulatory bodies are driving more widespread use of real-world data sources. And interestingly, it's not just for the generation of real-world evidence anymore. Certainly that's the sweet spot for the use of RWD. But what we're seeing now is the use of real-world data, real-world evidence in other interesting use cases, and in particular in clinical development. And so we're going to see how that's playing out. Number two, there's some terminology issues associated with single-arm clinical trials and the use of real-world data. Sometimes it can be confusing because some terms are used interchangeably. Some have subtle distinctions and so forth. So we're going to spend a little bit of time just level setting on terminology so that we have a solid foundation for moving forward. And then finally, the third thing I wanted to talk about was how there's really no one-size-fits-all approach to the use of real-world data sources in single-arm clinical trials. And there's different ways of characterizing how the applications of RWD in this context can vary. And I've got a little bit of material to help us understand some of these nuances and variations. So first of all, how are the use cases for RWD expanding beyond creation of real-world evidence? And, you know, a lot of it begins with the 21st Century Cures Act, which was signed at the very tail end of 2016. The overall thrust of it was to speed FDA approval of drugs and medical devices to get them into the hands of patients who need them most. It promotes innovative research designs for regulatory approval and has a lot of material in it, as many of you no doubt are already aware, on real-world evidence and different types of use cases for RWE in the approval process. One of the things that the legislation required was that FDA develop a program for real-world evidence and how they would be utilizing this kind of evidence in the regulatory process. 2 years after the legislation, they were mandated to put forth the framework for the RWE program, and you could see that this was published literally 2 years after the legislation was approved. In it, there's a single paragraph on the subject matter for today, non-randomized single-arm trials with external controls. And you could see in this that Just on the top end, they talk about how external, sometimes known as historical controls, are a possible type of control arm in an adequate and well-controlled study. They go on to talk about how historically this has been done by using the control arm from past clinical trials, but that in the future they could foresee more broad-scale usage of RWE. And then they conclude at the bottom by talking about some of the methodologic issues associated with using real-world data sources in this context, and the need to use advanced statistical methods such as propensity scoring. So all of these come into play as we start introducing this topic. Interestingly as well, a recent survey put out by Deloitte Insights here, I think it came out a month, maybe 6 weeks ago, entitled RWE Focus is Shifting to R&D, Early Investments Begin to Pay Off. And they do this survey every year, and interestingly you could see that they're saying that 94% of the survey respondents believe that real-world evidence is going to become very important in the research and development process by the year 2022. And in particular, they talk about a shift towards the application in R&D for things like supporting regulatory filings and augmenting clinical trials. And that's kind of a buzzword for what we're talking about here today. A little bit more detail in this, they asked the survey respondents specifically with respect to synthetic control arms, are they already using real-world data in this context? 29% said yes, which isn't that high, but when they asked them, do they expect to do so in the near future, literally 4 out of every 5 of them said yes, 82%. So this is clearly a huge growth area for the use of real-world data sources. And the question is why? So why does it make sense? And this is an article that appeared in Stat News, and really the headline captures it all. Synthetic control arms can save time and money in clinical trials. The benefits to the pharmaceutical industry are clear. By obviating the need to enroll study subjects into a control arm, you increase efficiencies, you lower the trial cost, you essentially cut your recruitment efforts in half. And it even goes beyond recruitment because it increases your retention efforts, as patients who suspect that they are on the placebo arm of a trial are often more likely to drop out. So the economics of it simply makes sense, and this is why this particular use case has garnered such attention. Okay, so I've talked about all the reasons why it's really cool and why we need to think about doing it. I haven't really talked about what it is, and so here's some of the fundamentals. So this is a depiction of the typical randomized controlled trial. We set this up as just a touchstone here. You have baseline, patients are recruited in, they're randomized at baseline into 2 different arms, active treatment versus control, and then they are followed over the follow-up period. And then changes from baseline to follow-up are then compared between the active treatment and control arms. In a single-arm trial, you have half as many patients, and you can see the arrows there are half as tall as the trial patients in the RCT. There's no randomization that takes place. They are simply enrolled in the study, all of them given active treatment. And again, changes from baseline to follow-up are assessed, but the design is weak because it has no control group, so you can't rule out that any kinds of secular changes over time in disease status are explaining the results that you are seeing, as opposed to being fully attributable to receipt of the active treatment. So what do we do about that? We develop an external control arm so that we supplement the single arm trial, which continues to be depicted at the top here, with an external control arm at the bottom, where we enroll external patients. They become our control group. The dotted line in between them serves to indicate that these are run as separate studies. But here, again, we have the ability not only to look at changes from baseline to follow-up, but to compare those changes between patients receiving active treatment versus control. And that's why the external control arm makes sense. So some additional terminology. Sometimes you'll see the terms synthetic control arms, and we flip back and forth here and there as well. We should consider these to be synonyms, although Craig and I are sticking with the external control arm terminology because that's more consistent with FDA usage. But there's two types. There's concurrent controls and there's historical controls, and these differ with respect to the types of data sources that can be utilized for them. So the single-arm trial with concurrent controls looks something like this, where we not only have alignment in terms of study time, that is to say the baseline and follow-up periods are the exact same duration between the single-arm trial and the external controls, but they're also aligned by calendar time. So this line down here showing the calendar time and indicating that these two studies are taking place concurrently demonstrates that everything is happening at the same time. So on the plus side, this ensures currency of standards of care, and it allows customization of study measures if you do this in the context of a prospective observational study. On the downside, there's feasibility issues of enrolling patients into a non-interventional study into this control arm. And if that is indeed feasible, some might question why you didn't do it in the context of an internal control arm, i.e., a traditional randomized controlled trial where you're randomizing patients to active treatment or control. Historical controls are more common for these various reasons, and they look like this. Again, there's alignment on study time with the exact same durations of baseline and follow-up, but there's misalignment according to calendar time, and historical controls happened further back in the past. So on the plus side, this broadens the range of potential data sources. You can use real-world data sources. You can also use placebo arms from prior RCTs. It's undoubtedly the case as well that retrospective data are more cost-effective than prospective data collection efforts. On the downside, and this is something you have to be careful of, the control data might become outdated if there are temporal changes from the time that those historical data were collected to the present. Changes in treatment patterns, biomarker status assessment, outcomes measurement, things like that. So that's something that has to be assessed in the context of looking at different types of historical data sources. So just some fundamentals on thinking about using an external control arm approach and evaluating different kinds of real-world data sources. These are sort of 4 questions that could be asked in sequence, and you need to have a yes answer to each question before you can move on to the next one. Any no answer is essentially disqualifying for the particular real-world data source. So question number 1 is the RWD source population similar to the trial population, in terms of demographics, in terms of geographic distribution, in terms of clinical characteristics of the patients? You don't want to be using, for example, a general health database when you're doing specialty care, say in the oncology or rare diseases space. You're simply not going to have the appropriate alignment between the populations. But if you get a yes answer to that, you move on to the next one, which is, does the database have sufficient granularity to enable us to replicate key trial eligibility criteria into the RWD source? In other words, do you have the information needed to overlay the inclusion-exclusion criteria onto the real-world data population? Things like the disease condition, how that's defined, how it's identified, as well as comorbidities, the current treatments and medication history. And of course, in the oncology and rare disease space in particular, things like biomarkers and specific diagnostic tests. If you get to a yes answer on that one, the third question becomes, can you do the same with the study endpoints? Can they be identified in the real-world data source? Things like treatment response and mortality. Also, do you have sufficient longitudinality in that database to match the trial follow-up so that you've got coverage over time without losing too many patients? If you get to a yes answer there, you move on to the final one. And that one is, do you have sufficient detail in the data to identify the covariates that are needed to do matching techniques? So demographics and clinical characteristics, treatment patterns, other kinds of non-medical factors, such as socioeconomic status can sometimes come into play for matching. So you need a yes answer to all four of these questions before you can even contemplate using a particular real-world data source in a single-arm clinical trial. And then finally, just wanted to get on this idea of there's no one-size-fits-all approach to doing this. So let's say we've identified a data source, we want to move on towards using it for external control. I'm going to lay out three different archetypes for utilizing real-world data as an external control. The first, characterizing this as an unrelated study that's used as a source of external benchmarking data. So here are the clinical trial patients. On the right-hand side, you can see that we know their gender. The colors are indicative of different types of characteristics. The simplest way of coming up with a control is just to go to the literature or maybe to some existing registries, have reports from them where we understand the natural history of the disease, some of the outcomes of care. In this instance, we have a control group, but it's sort of just used for benchmarking. So we don't necessarily know much about these patients. We don't know their characteristics, their color. We don't know their gender. So these are amorphous patients. They represent a control. And this is because there's no relationship between the trial and the external control data. There's kind of a brick wall in between them, as you can see. As we get a little bit more rigorous with this approach, we go to the second possibility, which is where we have an actual planned study as a source of quote-unquote trial-eligible external controls. And in this instance, we take the inclusion-exclusion criteria from the trial, apply them to the real-world data source, and come up with, as I said, an ECA consisting of trial-eligible patients. And these brown cones here are supposed to be, look like coffee filters. The same filters being applied to the control patients as were to the trial patients. This guarantees that we have some sense of, the gender breakdown and the characteristics of the patients. You could see that there's men and women represented here. There's also the same kinds of colors that we see with the trial patients, but there's really no guarantee that they're going to match. There will be, variations according to the underlying patient population in the real-world data source that might make certain types of patients crop up more frequently than others as we do this, as we do the filtering. But at the very least, we've applied the same inclusion-exclusion criteria and therefore have a little bit more confidence that we're doing this right. And in this instance, we then conduct the same types of analyses on the patients as we do in the trial. And then the final approach is where we actually have formal matching techniques that get performed. So again, it's a planned study. Instead of simply applying the filtering techniques from the trial, we actually use propensity scoring or other kinds of statistical matching techniques to actually look within the data set. For every single trial patient, we look for a match. And so what we end up with is a control group of patients who have a high degree of similarity on a one-to-one basis to each of the clinical trial patients. And so what this overall slide depicts is that, you know, there are variations according to how you go about doing the external control. And these variations are defined according to the connection of the external control data and the analyses to the trial. Everything from completely disconnected with a brick wall in between to completely connected with matching. And also they vary in terms of the statistical rigor in matching to the trial patients. Everything from no matching whatsoever to a little bit of similarity to one-to-one matching. And obviously, the more rigorous we can get with this, the better our results will be, and the more credible the analyses will be. So I'm losing my voice there, which is a perfect time for me to segue over to Craig. And I wanted to do so by announcing that Sineos Health is partnering with Craig's company, Concert AI, which is an oncology data provider, on external control arms in single-arm oncology trials. So with that, I will stop, and back to you, Sonia, to pass it over to Craig.
Yeah, so Dave just mentioned there that you know, Sineos and Concert AI are going to be partnering on external control arms. So probably good that I do a quick intro on who are Concert AI. Concert AI is a company that was founded about 4 years ago with the goal of applying artificial intelligence and machine learning to large data sets as a way to improve outcomes in life sciences, specifically focusing initially on oncology. We have access to about 400 sites, oncology sites around the country, covering more than 10 EMR systems. We have a nationwide representation for patients in the US, again, focusing specifically on oncology now. We get our real-world data from 4 different partners presently, 2 of whom we can name. One is Texas Oncology, the other is through the ASCO Cancer Link collaboration. And right now we're partnering and working with 3/4 of the top 25 pharmaceutical companies to implement solutions to accelerate, their oncology development programs. 19 of the top 20 life science companies are using one of either our RWD products, our enterprise AI solutions, or our services. Next slide, please. So Concert AI at present has the largest real-world data set in the industry. I mentioned already that we cover 400 cancer center practice locations around the US. Presently, we have 3.5 million unique patient lives covered. We're expanding every day, and by the end of Q3 and early Q4, we expect that to be over 4 million patients. We're covering all 50 states, and in terms of the breakdown of our data sources, approximately 90% of our patients come from a community practice and 10% from an academic. By practice, our practices themselves are 80% community and about 20% academic. And again, by the end of the year, we expect to be over 4 million covered lives. Next slide, please, Sonia. And the way that we implement our technology and data solutions breaks down into 3 or 4 main products. Fundamentally, we start with what we call our RWD 360 product, which is a structured data product. It's the largest footprint. It includes only the structured component of the record. And we link that to open claims where needed. We then also will reach into the electronic medical records, and we can apply a curation process either through NLP or automated techniques, or through use of clinical nurses and other practitioners to abstract data out of the records. And for that product, we refer to it as our Patient 360 product. In addition to the structured data that comes out of the record, we apply and create approximately 100 to 150, sometimes more, depending on the disease indication, variables that come from the unstructured or the free text portion of the record. We link that to closed claims, allowing you to see the healthcare resource utilization as well as all of the clinical and demographic characteristics. And finally, we have a product that we refer to as Genome 360. And for that product, the patients that are included are only patients who have access to a full NGS, a next generation sequencing panel. So going from structured to unstructured, to curated, all the way through to full NGS data, including all the biomarker data, positivity, negativity, and other, even down to the insertion locations, translocation issues, etc. when it comes to biomarker data for the patients that are in the Genome 360 product. Next slide, please. So when we think about how we do ECAs, there are numerous ways you can do them, and Dave has already mentioned a few, going all the way back to what I think is maybe one of the first ones in 2014, which used chart review. And a lot of clients who have done these in the past may have done them through chart review. That's a costly, an expensive, and a time-consuming way to run an ECA. And obviously, with the advent and the build of EMR data sets with the HITECH Act in 2010, there's now a wealth of data that are available that mean we don't have to go to chart review. We can pull data out of an electronic medical record. So I think when you start to think about how you would do an ECA, the first question is my trial or is my candidate product even suitable for an ECA? And so often what we're finding is that the FDA are amenable to studies where it would otherwise be quite cumbersome, difficult, or almost impossible to run a trial using the normal process. So those things are rare tumors. In our case, we spend a lot of our time working in oncology, and so typically what we're finding is that the types of trials that the FDA are amenable to implementing as ECAs are ones where the patient population is relatively small, the characteristics are hard to find, the prevalence is low, and otherwise it would take either a long time or it would require a significant number of sites, creating heterogeneity, or other factors that would lead it to just be otherwise unviable and non-viable to run a trial. And often what you'll find is that those studies are being implemented as single-arm studies anyway. And so the second piece, in addition to you initially deciding that your trial may be a candidate, is to have a conversation with the FDA or the EMA. We focus, or I'm going to focus quite heavily on the FDA because a lot of our work is done with the FDA. And you know, typically what's happened is the FDA has been the first approval body for a particular drug where it's been approved by the EMA. Oftentimes the FDA is the first one to have accepted it as an ECA-driven study. So having a conversation with the FDA, getting their buy-in, and ensuring that they are on board with the methodology and the approach that you intend to take. When you think then about how you would implement that, you also want to look for suitable endpoints. So there are studies where, for instance, the endpoint may just not be accessible in RWE. For instance, if a PRO is an endpoint, those can often be quite hard to find because they're not collected as standard of care. So things like progression-free survival, response rate, overall survival are the types of things that we find are quite useful and acceptable to the FDA when we're looking at external control studies. Second question is, are there data available for your study? You know, you can get data from chart review, you can get data from claims, you can get data from an EMR. But the question with some of these studies is the patient population so small that it's going to be impossible for me to find patients? So, you know, the next question you have to answer then is, where will I get the data from? And additionally to that, not just are the patients available, but do the types of endpoints or the types of criteria that you're using to include patients, do they exist in the data that you're looking at? So oftentimes, and what we'll talk about in a minute or two, is some studies in the past where the data that was used was perhaps not quite as suitable as it should have been because of missing this. Third question is, are the data suitable for regulatory use? So in addition to them being to having coverage of the parameters that you're interested in, can you apply full audit abilities to that data set? Can you satisfy the FDA that the data have been collected in a manner that's robust enough to guarantee that it's accurate? Does the abstraction process and other methods of acquiring the data satisfy 21 CFR Part 11 requirements? You know, we haven't yet seen the FDA require a GCP or ICH for these studies, but it seems like more and more clients that we work with are requesting some fairly rigorous implementations that require at least 21 CFR Part 11 electronic data capture requirements. And then finally, Can you do it in a way that's efficient and effective? We apply technology, including NLP and artificial intelligence techniques to identify patients who meet criteria as a way to filter through what is a large number of patients who might almost meet criteria but don't quite. So when you think about, how do I know if a patient has, say, a ROS1 mutation in non-small cell lung cancer? How do I ensure that this patient is metastatic? A lot of times that information is not contained cleanly in structured data, and it's only available through a human review or some other review of the record, and that review can be quite cumbersome. So how do you quickly get access to patients who meet the criteria that you need? Because as you'll see in the slides that follow, some studies that didn't do that did not fare very well in their approval process from the FDA. Next slide, please. So what have we learned to date? You know, the FDA have put out guidance. It's not binding yet, but it's indicative, saying how they expect ECAs to be utilized. And they can be used for one of three things, or multiple of three things. They can be used for evidence and support of a new product approval to satisfy efficacy or safety requirements. They can support labeling changes for changes to dose, indication, or even target population. Or they can be used to satisfy post-marketing commitments or analyses. So those are the three main use cases that the FDA have indicated, and they are in guidance that was provided. I think Dave had mentioned it in his slides, some of the early guidance and even updated guidance for industry with regard to how you might submit some of these studies. The other things that we're learning are about how the FDA perceive the data sources that are generated and how they're used. So fit for purpose data that's created specifically for the purposes of this ECA are preferred over off-the-shelf data. And there are multiple reasons for this. Off-the-shelf data tend to be limited in certain things that we're finding are absolutely necessary for these trials. If you think about the types of trials that are being done, again, there are rare tumors, rare diseases, very specific inclusion-exclusion criteria, the need for multiple lines of therapy preceding the drug that's being trialed in the intervention arm. They need things like ECOG. In some cases, you know, with liquid tumors and hematologic malignancies, there are composite identifiers of disease severity, like the IPSS, I think, that are often quite hard to interpret if you don't have the level of detail that you need in the data to be able to show that patients in the external control are a good match for patients in the trial. And so some of the comments that the FDA have made about trials that have been submitted as ECAs is that, for instance, the external control arm patient population are not well matched to the trial population. And in some cases, there's a strong perception that the trial population are a much healthier population due to things like missing ECOG in the external control arm population. The ability to identify patients who have died obviously is a critical component. And so how do you ensure that the death data are accurate? How do you ensure that you're capturing adverse events? Things like that you need to have full line of sight into as you build the data and create the data set for the study. Data auditability is important. So the ability to know where it came from, how accurately it's being collected. Stacking of data creates issues. So when we talk about stacking, you know, when you have a rare cancer or a rare disease, sometimes you have to go to multiple sites as you would with a trial, going to 5, 10, 15, even more sites to get enough patients. The same can happen with data. And when you try to pull multiple different data sources together that are created through different EMRs or different systems that are interpreted and analyzed in different ways, the ability to know that they are all representative of the same underlying thing is important to the FDA, and it's hard to prove that and show it unequivocally if you're pulling from multiple different data sources that are accrued in different manners. So the fit for purpose data from a single source that has full auditability is the preferred approach that we've kind of come to realize the FDA are looking for. And then methodological issues aren't uncommon. Things like immortal time bias, unobservable variables, things that you know, you can adjust for and you can accommodate if you think carefully about how you design a study. So requiring that patients have 6 months of follow-up creates biases. You know, patients who don't have 6 months of follow-up, they may have died. They may have been lost to follow-up because they went from a community practice to an academic practice that you don't cover. And so being aware of those things and designing the study appropriately so that you don't select patients who have either better or worse outcomes than the intervention arm is really important. Pre-specifying analyses, as much as it sounds like a very simple thing, in some cases that hasn't been done, and the FDA has been quite critical of sponsors who have submitted analyses that were not pre-specified. Limiting access to outcomes data, so making sure that, unlike with an interventional prospective trial where you don't have the ability to see the outcomes, with a retrospective analysis of an external control, it'd be quite easy for you to look at the outcomes of patients, and there could be perception of some kind of cherry picking or selection bias if you as a sponsor were to have access to outcomes data while you were selecting patients. As you think about Dave's slide showing one-for-one patient matching, you obviously can't match on outcomes, and in fact you don't want to know outcomes while you're doing that matching process, so that you can be assumed to have selected patients to suit your needs. And then the other thing that's important that people may not even consider is the fact that in a real-world population, they get treated according to standard of care and in a real-world manner, which means that they're not going to follow the protocol in the way that the trial does. The timing of scans, of diagnostic tests, of things like that, they happen when they happen. They don't happen at 30 days, at 90 days. They happen when the physician orders them. And so knowing that that is an issue and finding ways to deal with it methodologically is important. And it could be that you select patients who have those things done as close to protocol as possible, or you could adjust for them based upon some methodological adjustments. Next slide, please. Sonia, can you jump to the next slide? Yeah, so quickly, I'm going to talk about 4 success stories. I'll buzz through them. Alcensa from Roche got to market about 18 months early across Europe by using a synthetic control arm study, which they refer to as synthetic control arm, from an EMR database vendor of 67 patients as a comparative arm. So success story there, they got to market about 18 months early across Europe. Blincyto, blinatumomab, Amgen, and it's interesting to note the date here is 2014, and this was actually an initial approval for an accelerated approval based upon a phase 2 study. The full approval was granted based upon the implementation of external control arm. And again, that was 2014, before the 21st Century Cures Act was signed. Interestingly, that was one of the very first. AstraZeneca, Tagrisso, received approval, beating Loxo's Vitraki to market. Tagrisso got to market after 3 years. Vitraki, who started approximately the same time, took a couple of extra years. So you think about those 2 years of being in market due to the implementation of an ECA. And then finally, Merck KGaA, German Merck, in partnership with Pfizer and I think Lilly, brought avelumab to market for Merkel cell carcinoma. As you are probably aware, Merkel cell is a very rare tumor. I think it's only about 10,000 people prevalence, and they achieved US approval, accelerated approval, and conditional approval in the EU with historical control data from an EHR system in the US and from a German patient registry. And in that case, it was a natural history benchmark, because I think that Merkel cell carcinoma did not have a product that was approved for treatment. So, 4 examples there of success stories, and there are many more. I think if you look at across and outside of oncology, my math is probably not correct here, but I can count at least another dozen, or maybe even more in 2019 that were external control arms. And as we're finding at Concert AI, there's been a significant increase in uptake in the number of sponsors considering these for approvals, mostly for label expansions, but in some cases for an initial approval. Next slide, please. So there are a few examples of where it didn't go quite so right, and I'll talk quickly about 3. Selinexor, in August of 2018, was seeking an approval for third-line relapsed refractory multiple myeloma. It was rejected by the FDA. The Department of Epidemiology of the FDA commented on their study, saying that they did not have a protocol, and they didn't submit an SAP, a statistical analysis plan, before the study was to be approved. So the FDA was quite critical of that. It was also rejected because there was a lack of comparability between the RCT patients on baseline covariates. So things like ECOG, for instance, and I'll talk about this in each of these studies, in fact, the ability to get to ECOG is important because it defines the probability of survival. And in the case of an oncology trial, survival is the most critical component of the outcome. And then there were other issues like immortal time bias and selection bias, additionally missing data, and even index data issues. Erdafitinib for FGFR positive urothelial cancer in January of 2019. Again, methodological issues related to unmeasured confounders. Again, missingness of ECOG, missingness of death info. Selection criteria, because of the missing ECOG, the FDA commented that it indicated that there were probably significantly healthier patients in the intervention arm, leading to the probability of greater survival in that arm anyway. The other comment, which is it was somewhat unique to erdafitinib in my research, is that the FDA commented on the fact that the trial was in academic centers in Europe, but the RWE was from community settings in the US. So there was a perception that those studies were quite different. There was treatment misclassification, and there were also sample size issues. And then finally, for entrectinib, in July of 2019, this was for a ROS1 positive NSCLC population. Again, no study design submission to the FDA a priori. There was a generalizability issue because of the low rates of ROS1 testing. So in the trial, every patient was ROS1 positive, but in the external control, there was no way to ensure that all the patients were ROS1 positive. And there are differences in outcomes due to positivity. The study in the intervention arm, or the study arm and the external control arm, were deemed to be non-comparable. The sample size was too small, and there was missing this again in ECOG, but also with imaging info. Next slide, please. So this is probably the last slide that I'll cover here, but it talks about how do we implement ECAs. And when we think about how Sineos and Concert are looking at implementing studies, we start with a feasibility. As I mentioned, the ability to find the right patients is super important. Meeting with the FDA to discuss the protocol and the analysis plan, validating the patient and sample availability, and designing the protocol is very important, making sure you've got that buy-in. Building the data set and matching patients as the next step, gathering that baseline data, making sure that we provide data to sponsors such that they can see the characteristics that are important, but not the outcomes, to match to the real world sample, and then do the propensity score matching or other methods of matching so that we can have a one-to-one patient match there, and ensure that we have the sample size. As a next step, we go to data abstraction. We'll go in and make sure that we have full access to data records for the patients that are relevant, that the client wants us to use. Pull the data from structured components of the record, do the curation and the abstraction of the other elements that come out of unstructured, and then implement the quality control processes to ensure that it can satisfy that FDA or EMA review. And then finally, in the analytic phase, understanding lines of therapy, looking at outcomes, doing the sensitivity and subgroup analyses, and then even looking at follow-on studies for cost effectiveness and other elements of work that could leverage that same data set. We can even consider following those patients beyond the trial to see how their outcomes compare to patients that were in trial, or even adding patients as they become on therapy with a new drug to see how they are comparing in the real world. Next slide, please, Sonia. So just one example here, I think we're looking at a rare variant of lymphoma. And in this case, we're looking at it as a comparator to evaluate effectiveness, looking at progression-free survival and overall survival. The qualification criteria for this trial are that patients must have a confirmed lymphoma diagnosis. They have to have relapsed or refractory disease, which is an element that is not often available in structured data. It requires either AI or human review. And they have to have been treated with a BTK inhibitor, or in some cases a non-BTK inhibitor, but subject to specific lines of therapy and combinations of therapy. In this case, we're looking at a sample of approximately 500 patients, and that would cover 4 treatment cohorts. And in this case, it's going to take about 7 months to implement, and it's a 2-phased approach. We do our data abstraction and assessment. We make sure that patients match and that we have the appropriate sample. And then we go to phase 2, which would be the actual conduct of the ECA, the full data collection, including outcomes, and then the analytic phase. So I'm looking at the time here, Sonia. Although there are 2 or 3 other slides, I think we should probably just go straight to the end and let people ask their questions. So I'll hand it back to you.
Okay, thank you very much. Okay, I'm just going to go through the slides here and get to our last poll question, which is right here. I'm going to launch it right away so we can get to the Q&A. There it is in front of you. I'll give you about 45 seconds to do this last poll question. And the question is, to what extent will use of RWD sources for external control arms be an important consideration in your research moving forward? Please select one of the below. Not at all. I was just curious about the topic. A little, I can see potential for it, but not immediately. Some, I will look to implement this approach soon. Or a lot, I foresee widespread use of this approach. So please go ahead and cast your votes. Okay, it looks like the majority of everyone has done so. So I'm going to quickly go through it right now and close the polls and share the results. Okay, so there's the results of that last poll. And we have 2% that said not at all. I was just curious about the topic. 36%, a little, I can see potential for it, but not immediately. And then we have 30%, at some, I will look to implement this approach soon. And then 32%, a lot. I foresee widespread use of this approach. So thank you everyone for that. We're going to do the Q&A portion of the webinar right now. And if you have any questions, please send them through the questions chat box right there. I have already some questions brought in from the audience earlier, so I'm going to start with those questions. Here's our first question. Synthetic control cohort makes sense for a single arm trial. How about its application in a double arm trial where intervention arm is compared to real world control arm?
Yeah, there are variations on the theme here, and much of what we provided was simplistic, but there are hybrid approaches that get performed as well, where you have a traditional RCT with an internal control arm that gets supplemented with additional patients coming from external data sources. So that's definitely something that is another kind of variation on the theme, the type of thing that we can see in this regard.
Yeah, we've I'll answer this quickly. We've looked at different study designs. In fact, we've looked at augmenting a control arm where there may not have been sufficient patients. So if you think about trying to recruit 200 patients into a control, in addition to potential equipoise issues, sometimes it's just really hard to find the number of patients. So a couple of sponsors have asked us, can we provide additional patients so that they can more quickly recruit what would have been a trial that was stalled or was not sufficiently recruiting patients, so that they could make all the patients that they were finding intervention arm patients rather than control arm patients. So it's almost like they flipped over and had partial control through RWE and control through prospective. So yeah.
Okay, thank you very much, gentlemen. Here's our next question. I'm curious whether using RWD during clinical trials can assist with the racial disparities within clinical trials. I know it's a hot topic right now, and I would be curious on your opinions there based on your knowledge on RWD.
Yeah, I'll jump in and answer this one if you don't mind, Dave, and then you can add to it. The answer, I think, is yes. If you think about a typical clinical trial, they're heavily biased towards certain types of patients. You know, healthier patients, patients in areas that maybe are closer to academic medical centers, and oftentimes they tend to skew heavily towards one race demographic, so they underrepresent minorities. When you look at community practice data, you tend to find that there are a larger proportion of minority patients from different socioeconomic classes, and are more representative. So we've talked about this before, we being Concert AI and Sineos, regarding what does it do for the representativeness of these trials. And I think that you'll find that when you use an ECA, you have the ability to select. You could design a trial that matches the types of demographics that you would get from a clinical trial, but one of the benefits is you don't have to. You look to see what types of patients in the real world are using the products, and you can select those patients. So yes, it does have we tend to have a higher representation of minority and lower socioeconomic groups in community practice data that we pull from.
Without a doubt, I agree completely.
Okay, here is our next one. We have only 2 minutes left, and we have so many questions coming in from the audience. So let's see if we can get a couple more squeezed in. All right, here's the next question, David and Craig. Can you talk about data integrity and auditability of RWD to original source of data as would be expected in a clinical trial data?
Yeah, let me take that one again, if you don't mind, Dave. And so we at Concert AI have a technology platform that's 21 CFR Part 11 compliant, meaning that we know who touched the data, we know where it came from, we can replicate that whole process. We've been in discussions with the FDA about this. They've had a look at the way that we do it. They've commented that it at least is as good as the way that you would achieve it in a clinical trial. We know when you take a look at any specific data element that comes out of an ECA, you can trace that all the way back to the source. And I think given the time constraints, if people want to know more about that, they can follow up with either Dave or I, and we can go into more detail.
Yeah,
Okay,
I'd add is that the data provenance issue is very much in the news of late. With respect to the COVID-19 pandemic, there's been a rush of some publications leading to some embarrassing retractions once it was found out that the data could not be traced to the original sources. So prominent journals like The Lancet had to actually retract publications due to this.
Okay. All right. I'm going to squeeze in one more question. Okay. How can pharma companies leverage Concert AI technology for recruiting patients in clinical trials? Is that a capability that you have explored?
It is, and I think both Sineos and Concert have the ability to do that. We are working right now with Sineos on site selection and feasibility processes for clinical trials, both for prospective work and for retrospective work. So when you think about an external control arm, we, with our tools, have the ability to go into sites. We can do site burden analysis. We can do all kinds of things. And we can also facilitate and work alongside Sineos to identify where the patients exist, both for the prospective and for the retrospective portions of the analysis. Dave, do you want to add to that?
No, I think you've nailed it. And the only thing I wanted to add, given that we're at the top of the hour, is based on our second polling question, I was very encouraged to see that the vast majority of you seem to indicate that you'll be using external control arms in the future in your work. Only 2% said you were merely interested in attending the webinar, and hopefully you were entertained as well. But for those of you who do have plans for doing this, we do hope that this has been beneficial. Thanking you for your attendance, and I think you're going to put up, Sonia, our contact information.
Yes.
If you'd like to follow up, we'd be happy to engage further.
Okay. Well, thank you very much for those questions. We have reached the end of the question and answer portion of the webinar. If we couldn't attend to your questions, the team at Sineos Health may follow up with you or will follow up with you after this presentation. If you have any further questions, please direct them to the email addresses that's on your screen. There's one there for David and for Craig. Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event at that link and also share this link with your colleagues when they register for the recording as well. So I encourage you to do that. Now, please join us in thanking our speakers, David Thompson, Senior Vice President, Real World Research, Sineos Health, and Craig White, Senior Vice President and General Manager, Outcome Science with Concert AI. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, take care and bye for now. Thank you, gentlemen. Thank you, David. Thank you, Craig.
Thank you.
All right.
All right.
Well,
Bye, everyone. Bye-bye.